So esteemed delegates from the different parts of the country and also from the abroad, participants from the different universities and colleges, colleagues from the university. So good morning to all of you. So before I start my deliberations that redox remodeling of the redox neuron network in silkworm, insights from immunoproteomics and implications in protein writing, I must express my heartfelt thanks to these organizers, my colleagues from the department for giving me an opportunity to share some of our research findings since last few years, you can say 10 or 15 years, part of this study. So, esteemed chairperson, I already explained the importance of this silkworm in the context of not only in India, also. Yeah. Yeah. Thank you. So we understand this is a very important insect. So Sericinus insect, commercially important insect. So the objective of this study, I'll be explaining shortly. So this is before I start, briefly let me tell this is what our activities in our department that we use this insect silkworm and Drosophila model, visitor rat, cell culture based some studies, biophysical and computational studies, and we in our department mostly focus our study on the redox dynamics and immune response and epigenetics and this triangular relation, and they are very essentially physiological regulator of any organisms or cell or in any biophysical system. Mostly focus on the non-communicable diseases, natural compounds and metal-based hybrid molecules and therapeutic interventions, nanodrug and their delivery, and also the novel enzymes from the microorganisms and their therapeutic potentials. So we have a vibrant ecosystem, research ecosystems in our department. Also we collaborate with other the neighboring departments, sister departments, and also we have the Center of Excellence. So these are some of the research team, research ecosystems of our department. So today what I'm going to share, this is one of the area of the research we have been doing that in silkworm, that is the phosphor silkworm or Anthermylitis. So this is the redox regulations. So all of us should understand the redox regulation is nothing but the interplay between the pro-oxidants and antioxidants. So redox regulation is a universal phenomenon in diverse physiological functions, starting from the life to death. The redox play very eminent, prominent, important physiological role. The redox balance the essential component in development, aging, and also during the aging process. So from this we can see that the pro-oxidants, so there are two important players in this redox regulation. One is the pro-oxidants, where there is the reactive oxygen species or reactive nitrogen species, and the counterpart is the antioxidants. So usually the cell is to have a perfect balance between the pro-oxidants and antioxidants, and that is what we say redox balance or redox homeostasis. So when this pro-oxidants and antioxidant balance is hampered or tilted in favor of the pro-oxidant cell activity stress, that is called the oxidative stress. But virtually all cells have some sort of pro-oxidant generations during the process of respiration. We know that during the process of respiration we consume oxygen, convert it into water and energy. And during the process, some amount of free radicals or ROs are generated. So when these free radicals generated is overwhelmed, these are the balancing capability of the antioxidants. They build up in the cell, and when the ROS level increases, this is the normal ROS level. This is slightly towards the increment, and when it is the excess, that leads to the cell death. So it is essential as well as it is detrimental. So it plays a dual role, and you can say that is the coin of two sides. Now, the question is that, so if the ROS are detrimental, so why the process of natural selection, the process of evolution, why they have been selected and still in the diverse group of organisms? So they have essential role, and that essential role is that the killing of microorganisms. This is the first line of defense. The reactive species is the part of non-specific immune defense. The first line of defense, the phagocytic killing of microbes, it is mostly the ROS dependent or oxygen dependent killing. Oxygen dependent killing is a very important part of the first line of defense, where as soon as the bacteria or virus, whatever they try to enter the invade to the host, so there is oxidative burst, sudden rise in the oxygen consumption, resulting the release of the dramatic level of reactive oxygen species, which includes the superoxide radical, hydrogen peroxides, and so on, and they can kill the microorganisms instantly. So this is the first line of defense. So the pro-oxidants and antioxidants, that the redox couple, they are plentifully available in biological system. Not only biological system, also in the physical world or the biophysical world, there is ample importance of oxidation and reduction. So oxidation and reduction is a phenomenon, and two important players, the pro-oxidative players, there's the ROS, superoxide radical, peroxide, hydroxyl ion, and all those things. They are one group, pro-oxidant players, and we have antioxidant counterparts. They are proteins, vitamins, micronutrients, small molecules, and a vast array of natural compounds, starting from carotene present in carrot to elagic acid present in the spices. And we have lycopene in tomato, quercetin, curcumin, catechin, ethanol. So a good number of natural compounds across the vegetables, flowers have antioxidant potential. So today the story is all about the dynamics between these ROS and the antioxidants, and how our journey in silkworm, starting from simple redox regulation to proteomics, leads to the discovery of the protein that is transferred. So redox signaling is another important aspect. So starting from the anaerobic organisms, the bacteria and eukaryotic cell, from microbes to mass, the redox molecules or the redox components, the superoxide, hydrogen peroxide, and hydroxyl radical, they also have an important role in cell signaling, cell division, cell differentiation. So there is redox signaling, and there are redox sensitive transcription factors which are essentially regulated by the redox status of the cell. So redox signaling is an important phenomenon across the organism. ROS function starting off the life. I have told you that ROS play a dynamic role starting from the life to death. So as soon as there is a process of fertilization, the polysperm is blocked, and this polysperm block is associated with the abrupt release of hydrogen peroxide, and that leads to polysperm. So ROS starts and ROS functions starting off the life. Then come to the redox homeostasis. So redox homeostasis provides that is the balance, the perfect balance between the pro-oxidant generations and the elimination of by antioxidants. So that is what we say the redox balance, and this redox balance is essential for any physiological functions, whether it is the development, whether it is the differentiation, whether it is the cell signaling, aging, and finally the pathophysiological functions. There is no such disease in any organism which is not involved or associated with the imbalance of redox status or the dysregulation of the redox homeostasis. So no research is complete until it is not linked to the redox dynamics, because we are consuming oxygen, releasing carbon dioxide, and the process is an inherent process. The ROS has to be generated. So no physiological mechanism, no metabolic functions are without the involvement of production of ROS. And there are also group of antioxidants which remove them, which maintain them in the physiological level. So maintaining the physiological level of ROS inside the body is essential, vital for cellular function. When the threshold is increased beyond the threshold, it is deleterious. This is what the conventional or canonical redox regulatory network usually we see in most of all of the organisms. That is the source of generations of the different ROS, starting from endoplasmic reticulum. The mitochondria is a very potential source. Other cellular components, so by the different actions of oxidases, they generate the primary superoxide radical, which leads on to other free radicals or reactive surfaces, which is hydrogen peroxide. Hydrogen peroxide is very important molecule. It plays a dual role. It has antimicrobial action. It is also very important signaling molecule, and nowadays this small molecule is giving much attention for cell signaling, dormancy, and lifespan extension. So this superoxide radical, hydrogen peroxide, hydroxyl radical, all those things are being perfectly balanced by an array of antioxidant proteins, vitamins, and enzymes, and they maintain a perfect balance. And this is what we say conventional or canonical redox regulatory network. So our study, the story, the start here. So how the discovery of transferrin in this silkworm, they remodel this canonical or the conventional redox network, and we proved that transferrin is a protein with moonlighting function and play an important role in the redox regulatory network of this silkworm. Tropical thistle silkworm, Anthera mylitta, is a model organism for multidisciplinary research. This is a genus insect that produces lots of cells. The commercially important for textile industry. It is an important model to study insect-plant interaction, source of natural antioxidants, fibroin and sericin. Biological importance of the living biofermented bioincubator, model organism to study immune mechanism, antioxidant difference, aging and lifespan. Such an important organism this Anthera mylitta is. This is the life cycle of Anthera mylitta. So the eggs, then we have the liver and nerval stages, then the pupa, and the pupa has got a cocoon, and cocoon is the source of the raw material for the silk production. With the cocoon, we get the silk. Silkworm is not only of biological importance. It is a lot of studies have been carried out on interface of biology and physics. So silkworm is a model organism for interdisciplinary research, and the people have discovered the silkworm cocoon has the ability to transform the solar light into electric current. And this is an equivalent model that solar light is converted into electric current is converted into the signal transduction pathway by which the pupa emerges out. So what is the cues, what is the environmental cues, how the pupa knows that why it should come out. So it is getting the information from the solar radiation, that is converted into electric current, the electric current stimulates the biological signal transduction pathway, and it comes out. And lot of paper has been published in scientific reports that silkworm, the pupa, and also its covering the cocoon can be a future biocompatible solar cell. So it has also ample importance in the biophysical studies and also in terms of biology and physics. So this is how this silkworm cocoon is made up of a protein, fibroin and sericin, and it maintains a gradient of salt. So when it absorbs moisture, the gradient of salt and the thickness that creates the voltage, and that voltage or potential difference is converted into electric current, and they induce the pupa to certain signal transduction pathway by which it emerges out. Some enzymes is activated and emerge out. Very important, beautiful model and beautiful concept also. Now we come to the real story that redox regulatory network or redox modulatory network with the system biology approach, that is the silkworm, and we have used the techniques of bioinformatics, epigenomics, immunoproteomics, and blended to the discovery of the protein transferrin, which has got the moonlighting functions, multiple functions, single protein having the multiple roles. So this starts with the observation that catalase is a very important enzyme in the redox regulatory network. Very important enzymes, very respectable enzymes, catalase highly conserved from microbes to man. To our surprise, that when we are doing this study on the silkworm, a particular tissue that is hemolymph, insect hemolymph, we did not find the catalase activity despite our several attempts. So initially thought that we have something wrong in our protocol, but when we could not observe this catalase activity, we give a different look into this story. So we tried to validate that catalase activity is not available, not detectable in this level, and it is available in other tissues in the same organism, same insect. So hemolymph is not having measurable catalase activity with the available methods we have used to detect in other tissues. That's the beginning of the story. So no catalase activity in hemolymph, but it is there in midgut and other tissues. So catalase is an important enzyme, I have told. And these findings we published in very important journal, International Journal of Insect Science, that there is no cartilage. First time we reported that no cartilage activity in the hemolymph of this species. Then we looked into the matter why this such tissue-specific function of redox regulatory network is different. Why cartilage is not possibly present or not below the level of detection, and that we look into the different aspects. We tried to give some inhibitor of the cartilage to see what happens to other organs where cartilage is there. So we could observe that H2O2 very important molecule in this insect, particularly regarding the dormancy, development, and lifespan extension is associated. And we observed that when we injected the cartilage inhibitor, the other tissues they modified the redox regulatory network and somehow accommodated our physiological ability to survive. So we come to a conclusion that H2O2 is an important molecule in this insect, and possibly cartilage has some evolutionary role to play, not to scavenge the hydrogen peroxide, particularly in the hemolymph, to maintain a physiological level. This is what the hypothesis we draw at that time. And we published this paper in another important journal, that is Entomological Science. Then we had a different objective. When this was a project funded by SCLB, Government of India, our objective was to how to improve the quality and quantity of silk production by giving a known antioxidant inducer. So that was the objective. So we have selected ascorbic acid, the vitamin C, and also the glycine, which is an important component of the silk. So we are interested to improve the quality and quantity of the silk production by giving the glycine and ascorbic acid. We did the experiment as a part of our objectives. So we sprayed ascorbic acid and glycine to the foliar and the leaf, and we could observe that oxidative stress is reduced, antioxidant get boosted, biophysical parameters or trait of the cocoon silk, percentage of the silk, and the tenacity of the silk also improved, and all the antioxidant enzymes are also being elevated. So that is the objective, how to improve the quality and quantity of the silk by supplementing hydrogen by ascorbic acid and glycine. So we could partially achieve the objectives that the yield of quality of quantity of silk was improved by supplementation of ascorbic acid and glycine. So that was our objective. But the other observation that this is all those expression of P53, superoxide mutase, and PCA, proliferation cell nuclear antigens, all these important vital molecules are elevated in the cell wall by supplementation of ascorbic acid and glycine. But what is more important and more interesting to us, this is tissue-specific protein expression in hemolymph, which was not a direct objective. Just we are trying to understand that in response to ascorbic acid and glycine supplementation, what happens to the profiling of the protein. So we observed that initially when we did the experiment, that 2 microgram of protein, 5 microgram of protein, the lens were spilled off. Such a huge expression of protein, and this is particularly in hemolymph, not in midgut, where catalase is present. In hemolymph, there is highly upregulated in response to supplementation of ascorbic acid and glycines. So we become more inquisitive to understand what is the possible mechanism. Then we try to understand whether the same phenomenon is occurring in the normal tissues without supplementation of ascorbic acid and glycine. So we not only did it in the larva, also we did it in the pupil. So in the pupil hemolymph and larval hemolymph, we also observe that without any trigger, without any supplementation, that particular protein, 75 kilodalton protein, is being abundant. It is expressed, overexpressed. That is further interesting insight that so it is not a protein induced by ascorbic acid glycine, it is inherently upregulated during the process of development. So we try to understand it might have some role in the process of development. then we give more focus to identify this unknown protein. This protein, but at that time we didn't have this facility to identify the protein. So the person concerned, the student, scholar concerned, he took this protein band, he went to Institute of Bioinformatics of Bangalore and stayed there for 15 days and did this mass spectroscopy. And after this mass spectroscopy analysis, that we could identify that protein is a transferrin. Transferrin is a protein that was reported from microorganisms, reported from insects. Transferrin is a protein present in almost all organisms, but the role played by transferrin is multifarious. It is context dependent, may vary from individual to individual, species to species, but we for the first time reported the 75 kilodalton protein by liquid chromatography coupled to mass spectroscopy as the transferrin. Then at that time, to identify the protein transferrin by mass spectroscopy for a single sample was 35,000. Then we thought that why not to develop a specific antibody so that we will be able to detect the protein by a targeted proteomics, that is either Western blotting or immunohistochemistry. So we thought of purifying this protein and developing the antibody. So the purified protein, we tried to develop the antibody, and this antibody against the transferrin. So transferrin that is primarily involved in iron homeostasis across the different animal kingdom. So the first 20 amino acid sequence of this transferrin from the silkworm Enteramyceta, and we started developing the antibody. Also, that we tried to make a homology among the different insect groups. That what is the percentage of the homology in the different insect group available from the database, and we also got a good percentage of homology in the different organisms, starting from the Bombyx mori, other silkworm, and also the other insects. So we did a bioinformatics analysis and we could establish that it's a good homology is there. Then we thought to develop the antibody to minimize the cost of detection of transferrin in the different organisms, and also to develop a product which may be another important objective. So we did this not in our laboratory, so we had to customize this production of antibody in the MGNX India Private Limited, and we could successfully raise the antibody. So raising antibody, developing antibody is not enough, it's not complete the story. We need to validate the antibody. So the antibody efficacy was validated by Western blotting, so we could detect the transferrin, the range of tissues starting from hemolymph, midguard, silk gland, fat body, so in larva and also pupa. So it reacted, and also not only reacted to this Enteramyceta, also cross-reacted with the other species like Spotoptera, Drosophila, also including the liver of rat. So this is very important, successful antibody, which is polyvalent antibody, but has the cross-reactivity to detect the level of transferrin, the range of organisms starting from insect to mammal. We also validated its efficacy not only through Western blotting, also validated through immunohistochemistry and also through the immunocytochemistry. So this is the site in the hemolymph, which exhibits the good amount of transferrin is accumulated surrounding the nucleus. Then we come to a conclusion that possibly transferrin has some role in antioxidant protection of the insect, because it is abundantly expressed in hemolymph, a tissue which is devoid of measurable amount of catalase activity. So nobody will believe until unless we prove it. So we try to validate functional validation of transferrin as an antioxidant. So we did some experiment. So we did the experiment by injecting the known pro-oxidants like ferrous sulfate, ferric chloride, ascorbic acid, hydrogen peroxide to the different stages of the larva to induce oxidative stress and to observe what happens to this level of transferrin. So when this ferrous sulfate, ascorbic acid, hydrogen peroxide, all inducers of oxidative stress were injected to the larva, this particular plant having this particular protein is upregulated, gives an direct evidence that it has very important role in mechanism of redox regulatory mechanism means in providing the antioxidant protection. Not only that, we also observed in the normal cell phone, not during the normal development, that particular protein is upregulated with reference to the development. The different level stages, third instant, fourth instant, and fifth instant. So during the process of development also, this particular protein is upregulated. So gives an idea, a clue that this protein must have some role in the process of development. Definitely, because in the process of development, third, fourth, and fifth, the advanced stage of the larva has got the accumulation of the enhanced production of transferrin protein, so it has got some role in development. So transferrin is expressed, upregulated in response to oxidative stress induction. It is upregulated during the process of development across third to fifth instant of larvae. So it says, or it strongly suggests it has some role in development and also in redox regulatory network. Not only that, we tried to prove our hypothesis with another model insect, Drosophila. So we treat the Drosophila with antioxidant inhibitor. One is GSH synthesis inhibitor, Bithynium sulfoxamine, and there is ferric chloride and ferric ammonium acetate, induction of oxidative stress. So we could observe that the reduction of GSH, increased amount of RBS, and transferrin is upregulated, and 4-HNA, another marker, 4-hydroxynonenal is also upregulated, and one important marker, which is the marker of apoptosis, iron-induced apoptosis, that is gamma-glutamine cyclotransferase, is also upregulated. Here also, with response to efficiency and ferric ammonium citrate induction, transferrin is down-regulated. Again, gives a convincing evidences that transferrin has some role in the process of apoptosis, and it can be used as a protein during the process of regulation of apoptosis. So accumulating evidences indicate, suggest that all these functions are associated with the metabolism and dynamic of transferrin, starting from antioxidant protection, redox regulation, development, and also apoptosis. Then came a very important study that this particular insect has got a life cycle, and particularly it has a dormancy period. That is what we say diapause. Diapause is a pause in development, is a arrest in development, and particularly during the stage of the pupa, that sometimes it remains 21 days, sometimes it remains 130 or 40 days. So such a huge extension of the lifespan. So one life cycle is completed, the pupal cycle only in 21 days. The same organism in a different cycle remain to extend up to 130 days. Huge amount of life extension. And we could prove that this is associated with the dynamics of hydrogen peroxide. Hydrogen peroxide is a hormetic trigger, H2O to low hydrogen peroxide, and enhance oxidative resistance during this life cycle extension or the process of dormancy and aging. And we published this paper, very important journal, Zoology, that indicates that low hydrogen peroxide pool with a strategic antioxidant protection associated with the induction of dormancy and hence oxidative stress during the extended lifespan. So hydrogen peroxide has got the good role over here. Not always is a pro-oxidant, rather it has a physiological signaling role it has. So with all these convincing evidences, we came to a conclusion that the protein transferrin reported first time in this insect species exhibit a moonlighting function, starting from its primary function, that is the iron homeostasis, additional functions, redox regulation, larval development, innate immunity, antimicrobial action, aging, dormancy, and also very important biomarker for cancer, particularly oral cancer. And nowadays it has got a lot of therapeutic applications in neurodegenerative diseases or Alzheimer's, particularly Alzheimer's. Then our first observation that catalase was not expressed in a measurable quantity in people. So we try to understand what may be the possible disease. So the loss of catalase activity in this tissue is a chance, is a choice, or it's a evolution-driven phenomenon. And such downregulation of gene is it related to epigenetic gene silencing, specific epigenetic gene silencing. We did some experiments and we had some information that particularly in honeybee, other insects, that the changing the diet could change the sex. Changing the diet could change the sex. The same organisms develop into queen or to worker when the diet is changed, and this is reported that this is epigenetically triggered. So epigenetic trigger, if it is a reversible phenomenon, yesterday we have listened very good talk on social transformation of epigenetic. So epigenetic is a reversible phenomenon, and DNA methylation is an important epigenetic modulator. DNA methylation, without changing the sequence of DNA, that cytosines and other base gets modified, and this is what is called the epigenetic, means how the environment influences the genetic makeup and leads to different type of phenotypes. So we have the information, and this is the basic mechanism of DNA methylations. A group of enzymes, the DNA methyltransferases, which write this means that they can induce epigenetic modifications or DNA methylation. Then a group of proteins are which are the marker, means when there is methylation, those proteins should be there, and a group of proteins are there which erase it, means they can reverse it. So it's a reversible phenomenon, and this is the basic mechanism. And so sometimes it was reported that the absence of a particular protein in a particular tissue, maybe it is the DNA methylation dependent hypermethylations and gene silencing. So those informations were available to us. So this we did an experiment, and we had also done some experiments on the rat, that in the rat, when the rat was made hypothyroid, catalase was downregulated, and this catalase downregulation was associated with hypermethylation of catalase promoter, which already we have published earlier. So based on our own experience, based on the evidences available from the other group of workers across the globe, we did some experiments and were also very successful in search of epigenetic signature. We took the two tissues, one tissue where catalase is there, one tissue where catalase is not there, on a hemoglobin and then with that. And we could establish that some of the signature molecules like DNA methyltransferases, 1A, 3A, methyl binding domain protein, and GAL45, these proteins, they are differentially expressed in this tissue where catalase is available and where catalase is not available. Gives a clue that absence of catalase activity possibly associated with epigenetic regulation. And we did another experiment, and we had an information that promoter of catalase is a catalase promoter. So finally we did that cat enhancer binding protein CB beta, which is a transcription factor regulating the catalase gene, is also differentially expressed. So all those experiments strongly suggested that downregulation of catalase in hemolymph is an epigenetically driven phenomenon, possibly to maintain a physiological level of hydrogen peroxide in this tissue, which is a very important role in the process of development, dormancy, and life extension. With the discovery of transferrin, with the discovery of epigenetic gene silencing of catalase, we remodeled the redox regulatory, conventional redox regulatory network with a new player, that is transferrin. So transferrin is a new player in the redox regulatory network of this silkworm species. We published this paper in comparative physiology and biochemistry. We developed the antibody. So antibody is also commercialized, commercially available, which have been released by the Honorable Minister of Applications in presence of the Honorable Vice Chancellor. So it is commercially available. So this is what my message that biology to bioeconomy, with the technological intervention in animal science research, with the tools and techniques. Your take-home message: biological system, a treasure house of novel and fascinating phenomenon. Research is not a linear process; it is iterative and interdisciplinary. Starts with the critical observation analysis, and everything lies in your brain. Brain is the real laboratory. Sir Christian Push. So I must acknowledge the contribution made by the research scholars in different times, particularly these three research scholars, Dr. Venus, Dr. Monica, Alpuna. They started this work long back when I was in Northwest University, Baripada, and they have substantially contributed to accomplish all those studies. So I must and to all of him and also the funding agencies and all the scholars who are involved in this study, and particularly for the DST Government of India, DBT Government of India, and DST Government of Malaysia. Thank you.
Thank you very much, Professor Dr. Jagannath sir. The talk is open for discussion. Yeah, we can take up. Okay, so that's a wonderful talk of question is just on one part. So you have already shown mastery, right? So you are already saying that it is almost all the page means the markers that you showed is an STS page. So the question is, transferring can also behave in oligomers. Like the one, because why I'm asking this is because you're getting such a huge amount, means the organism is really making it a lot for it to support. So is it like a monomeric protein or is it still you doing HPLC?
Only one thing we have observed that the same problem we are facing for its purification is heavily glycosylated. Okay, so when we treat the transparent with deglycosylated enzymes, then the band is slightly shifted. But we are not able to get the crystal structure. We have tried so many times, but we are not able to get the crystal structure because of heavy glycosylation. So we are trying a different way to get the crystal structure. Once the crystal structure is there, then we can get all those.
Yeah, but can we like check it in a HPLC or a native page or a
native page we have tried.
Okay, but that is, it shares.
Yes. LCMS we have tried. LCMS it gives a good band, specific band.
And in the LC it is giving
yes. Yes.
So that's what because it's a huge expression.
Yeah. Huge expression.
Yeah.
Initially when you did the experiment, 5 micron protein the range is still up.
Thank you. One question, last question.
So thank you for a very interesting talk. So you mentioned that this mechanism is involved in maintaining diapods. So I was wondering, at the end of diapods, when the insect has become active again, is this whole thing reversed? And if so, how is this reversal set in motion? Do you know anything about it?
So diapod is a natural phenomenon in this insect. So it is mostly triggered by environmental cues, that is the temperature, availability of the food, and also some genetic studies. The Dellinger is a authority in the diapod. They have done a lot of studies. But we are not able to understand that why this particular organism has got a life cycle. When it can complete the people within 21 days, but sometimes it's to even 130 days. So we're trying to understand what the possible, the mechanism of the different proteins that play a role. And we have so far one observation that hydrogen peroxide is possibly a triggering component. It gives a resistance during the process of diagnosis. But if we can revert it, or we can just there are some people they have done some study, but abrupt disruption for diagnosis by giving some agonist of some proteins that it comes out, emergence of this paper comes out. But another thing is that since that the two other reports of the scientific report they told it is the solar radiation. So it may be the diurnal variations of the length of the daylight that giving an optimum signal to come out from that. So it's a very interesting interpretive questions, and possibly it's a very important evolutionary driven phenomenon need to understand. We know then only we can also give some solution for the aging process also. Reversal of aging.
But the overexpression of transferrin stops at the end of
So we are trying to clone this and trying to disrupt these things by inhibitor or by genetic mechanism. Then we are interested to see whether that. I am very sure that downregulate the transferrin, there is a huge disturbance in the developmental pattern that we are expecting, but so far we have not conducted such experiment.
Okay, thank you.
Okay, thank you, sir. Sir, I have a question. Sir, I just want to know if so this oxidation process, does it affect the production of silk, silk quality?
Yes. The silkworm has got three different host plants: Terminalia arjuna, Terminalia tomentosa, and Soria robusta. So people have observed that the mortality is high in Soria robusta, but the silk quality is better. So less number of larvae survive, which gives a robust cocoon, very good quality of the silk. So we analyze the deep compositions by PIXI, very sensitive technique. We observe that the salif Soria robusta has got a very high amount of iron. So possibly the enhance of this particular protein in fifth instance, it may be an antioxidant protection to iron induced oxidative stress, which is inherently consumed to the feed. So, but the enhanced oxidative stress reduce the larval fitness and also the quality and quantity of the yield of the seed.
Sir, then what a farmer can do?
Yes, did an experiment in the farm also in the sericulture field also. By taking this farmer, 2-3 farmers we have taken the beneficiary. So we spread the ascorbic acid, we spread the glycine, and there is a tremendous output of this in terms of the both quality and quantity of the seed. That also we have to do. And we have patented 5%.
Okay, thanks very much. Now we are honoring our plenary speaker, Dr. Dennis, on behalf of the organizing.
May I request today's chairperson, Professor K. Murugan, to felicitate today's first speaker, our plenary speaker, Professor Jageshwar Darpar from Department of Biotechnology, Utkal.
Nine water presentation and also two more presenters are also interested to have a presentation in this session. So that the first presenter, Mr. Shivani Mukapatra, is here. Please come. You can present your paper entitled Incorporation of Submergence Tolerance and Durable Microbacterial Blight Resistance into Popular Rice Variety. Yeah, podium. Yeah. You are welcome, madam. Good morning.
Good morning. Good morning. I'm Shivani Mahapatra. Today I'm going to talk about my topic, that is incorporation of submergence tolerance and durable bacterial blight resistance into popular rice variety Rani Dhan through molecular breeding. This work was conducted at National Rice Research Institute, Vidyadharpur, Cuttack. As we all know, that rice is one of the staple food crops throughout the world, and India is one of the largest rice growing nations in the world. As the global population is rising at a very alarming pace, by the year of 2030, it will need around 20 to 30 million tons of milled rice per decade. Therefore, in future, it will be an extremely challenging task to fulfill the gap between the demand and the supply. Here in our work, we have improved Rani Dhan, a very popular rice variety in eastern India, and that is also a late maturing variety, and we have made it tolerant to different biotic and abiotic stresses. We have targeted it with submergence tolerance because submergence is a very submergence in eastern India. Flood is the major problem for around 16 million hectare lands. The bacterial leaf blight disease resistance is our next target, as this disease can reduce the crop yield up to 60 to 80%, and this can limit the crop yield also. So, for this experiment, first we have done the hybridization. This work was started during the wet season 2016 and ended in the wet season 2021. The molecular marker interrogation backcross breeding program was done to transfer proposed 4 genes or QTS into Rani Dhan variety. During the backcross generation, direct markers were used for sub-1A, and markers closely linked to the genes were used for XA13, XA5, XA13, XA5, and XA21. The 2 donor parents, Swarna sub1 and Swarna mas, were first screened for parental polymorphism and then validated for presence and absence of the target genes and QTS. This is the gel picture for the parental polymorphism. After completion of the cross with multiple parents, the first backcross was done and 396 seeds were produced, and further foreground selection and further evaluation was conducted, and we have found 11 plants with our target desirable genes. After that, the BC2F1 generation, we have found 321, and BC3F1 generation, we have found 286 plants. And in the end of BC3F1 generation, we got 12 plants with the maximum similarity to Rani Dhan, and those plants were cell-bred. And we have got 1270 seeds for further evaluation. After that, those 1270 plants were screened with the markers sub1A20, sub1BC2, RM8300, XA5, XA13, and XA21. And only 4 plants carrying all the desirable genes were found. These are the representative electroprogram for BC1F1. This is for BC2F1. This is for BC3F1. After that, the selfing of those 4 plants, a huge number of plants were produced in BC3F2 generation and were screened, and 7 genotypes were selected for selected. 4 of them carry all the 4 desirable genes. 3 of them are with different gene combinations. those 10 plant set was carried forwarded. This is the gel electroprogram for BC3 F2 generation. After that, we have validated the submergence and the bacterial blight resistance. First of all, we have done the test genotype was exposed to complete submergence stress of 14 days. The regeneration ability was validated, and firstly, the regeneration ability varied from 70 to 70.6% to 94.5% in the all 7 pyramidal lines, and carrying sub 1A. And while the donor pack sub 1, Swarna sub 1 showed a regeneration ability of 87%. This is the graph which is showing the first 7 are 7 pyramidal lines. Then the Swarna sub 1 is the donor parent, and the 2 BLBs donor parent and the recessive parent. Here we can see one of our 7 pyramidal line is showing highest regeneration ability. While the susceptible variety is CR 800, or it is also called Swarna Mars, and Rani Dhan did not show any regeneration and died as a result of 14 days of submergence stress. After that, we have validated the bacterial leaf blight disease. Then 7 pyramidal lines included 4 homozygous plants, carried 3 bacterial leaf blight resistance, and the 3 parents were evaluated during the wet season 2020 and 2021. The bacterial leaf blight disease lesion length was measured after 4 weeks of XOO strain inoculation. This is the diagram. Here we can see the lesion length of one of our 7 pyramidal lines. The lesion length was shorter in the donor parent, and this could resist the bacterial leaf blight pathogen attack, which showed a mean of lesion length of 1.93 centimeter. However, the recipient parent Rani Dhan exhibited a longer mean lesion length of 9.81 centimeter. The mean lesion length observed in the derived lines varied from 2.14 to 2.25 centimeter. The results clearly indicated that the pyramidal lines with bacterial blight resistant genes exhibited higher resistance compared to the recurrent parent, that is Rani Dhan. After that, we have evaluated all the 7 pyramidal line and the parent in comprising in agromorphological attributes. According to the 2 years evaluation data, the recipient parent showed a mean grain yield of 5.89. This is the diagrammatic representation. Here we can see the yield of our 7 different pyramidal lines, the 2 donor parents and 1 recipient parent. However, all test entries carries the 4 target genes showed a higher grain yield than the popular variety Rani Dhan. The pyramidal line, the first one, showed the highest grain yield, that is 7.17 ton per hectare, followed by 6.99 ton per hectare, 6.84 ton per hectare, and 6.76 ton per hectare. All pyramidal line exhibited higher yield than the recurrent parent Rani Dhan. The best pyramidal line, CRSB 159, 87, 69, 942, showed a yield advantage of 21.73% and 9.22% over the recurrent parent and the bacterial blight resistant donor parents respectively. So these are some of the different parameters we have taken, that is for plant height, days to 50% flowering, panicle length, panicle weight, number of panicle per plant, number of grain per plant, spike fertility, and 1000 seed weight. After this experiment, we developed pyramidal line carrying the submergence and bacterial blight gene were similar to the recipient parent in terms of 9 agromorphologic traits. These pyramidal lines will be highly suitable for flash flood affected areas of eastern India. Thank you.
Congratulations. Congratulations. Only 1%. If you want. Yes, please. You are asking question? You are giving my, yes. Or since there is no question, again we are congratulating you. All the best. Nice presentation. Okay. Yes, sir. Yeah. Here, sir. Yeah. Who is giving? You can take your side. So next speaker,
Now,
Mr. Santosh Kumar. Okay, what's your name? Oh, you want to. But you are come, I can't use a person. Okay.
Now I would like to invite Dr. Santosh Kumar Sahu for his oral presentation.
Keep the mic here. Keep like this. Suppose, huh? You are an aeronautical engineer. Come, quick. So, walk fast. Okay.
Can we change the slide? Good morning, everyone. So, my work is on model biomimetic membranes. So, model membranes are essential for research because, next slide, please. Next slide. So, because the purification of membrane is a tedious job, it needs machines like ultracentrifuge. So, ultracentrifuge is a very heavy and expensive machine. So, it's all average for these labs can't have this machine. So, we need to prepare membranes synthetically. So you have to take the lipids and follow the procedures to get the. So liposomes are, they look like this. So they can be of various types depending on the number of membranes and their size. So liposomes are model systems for different research work such as gene delivery and translation of eukaryotic cells, targeting of drugs, then they are used as model system for study of structure, function, organization, and dynamics of proteins, membrane proteins and lipids. So they are also used as carriers for enzymes, elation therapy in removing heavy metal toxicity, and they are also used in drug resistance in pathogens, and they are also used in tumor therapy. So there are different methods. Several methods have been used for formation of liposomes. So one of them, all these methods are little expensive. So they use expensive gases and exportants. So we devised a very handy method to form liposome in laboratory. So this is a handmade generator, and we use these conditions, and these are copper electrodes. So we take the lipid deposits on the copper electrode, then we put both the cups like this, we seal it, then pass electric current. So the current should be this one. So in 2 hours, we get liposomes. So So we call these giant unilamellar vesicles because they are larger in size. So the average size is around 100 to 800 micron. So after they are formed, we did the testing to know that they are unilamellar. So unilamellar vesicles are more favored because they have single membrane. So this is an assay, it is a fluorescence based assay to check that when they are having only one membrane, not multiple membrane like this or like this. So their average size is 80 micron. So depending on the type of liquid we take. So the cost analysis shows that the price is coming 0.11 pound, that is 15 to 20 rupees for preparation. So they have several advantages, but one of the most disadvantage is that we cannot directly incorporate proteins into this glycosome. So for that we have to follow an indirect method. So there are several applications of liposomes. So I am going to tell whatever we have done in our lab. So in our lab, we focus on a study of drug resistance in bacteria. So these are the group of bacteria escape. So these bacteria are, mostly the drug resistant varieties. So they resist to different drugs. Most advanced group of drugs, and it's very difficult to treat. So one of the important aspects we use this membrane to study is to analyze if any phytochemical is lysing the membrane. So if the phytochemical will lyse the membrane, then the assay will be like this. This dotted line shows there will be decrease in fluorescence. So if the fluorescence decrease is only 50%, then the basic is intact. So if it comes down, more it comes down, the more membrane trophic the compound is. The more larger is the lysis. So this membrane trophic phytochemicals, they are enhancers of antibiotic efficacy because they permeabilize the membrane. So we did this experiment on live bacteria to show that the IC50 concentration of these antibiotics were decreased in presence of the membrane trophic compounds from gymnoacid based. So to publish this work in ACS Omega, where we took the gymnoacid based, extracted the phytochemicals, then we treated the large unilamellar vesicles with these phytochemicals, and we found that the phytochemicals permeabilize the membrane and they result in enhancement of antibiotic permeability into the bacteria that resulted in the enhanced efficacy. So in summary, membrane trophic compounds they enhance the bacterial membrane permeability, and they could be potential prodrugs or used as antibacterial or adjuvant therapeutics. So one more important aspect we are dealing with is to study how resistant a bacterial membrane is towards a membrane-like antibiotic. So there are many antibiotics which target the membrane, but it depends on what type the bacteria is. If it is a resistant type of variety, then the membrane lysis is lesser. So here you can see that the dotted lines don't come much below. So here we use two varieties of Staphylococcus aureus. One is antibiotic sensitive and another is antibiotic resistant. You can find the difference between the two. So this is a dose dependent analysis, membrane lysis, and this one will be time dependent. So in both cases, there is a striking difference between the two states. So here, we also use some more experiments to validate our hypothesis. So we did the PAP assay. So here we prepared the artificial membranes. So these membranes contain a fluorescent lipid which gives this red fluorescence. But when we do the bleaching, so this fluorescence, I mean, the fluorescence is erased here, and after some time, some 10 to 15 seconds, the fluorescence recovery takes place in this strain, so which is not resistant. But in this resistant variety of strain, so you can see that the recovery doesn't have any effect. So actually here we use some more bacteria to validate this hypothesis, and we found that in the resistant variety, the recovery of this fluorescence is very slow. So this one is the resistant variety of SRS, and this one is the sensitive variety of SRS. So we also did the PET assay to validate our hypothesis. So here you can see that in drug sensitive strain, so there is a the mobility of the membrane is higher. So this is an assay to show how mobile the the lipids are. But in this case, the lipids are very immobile. So here we also did some electromicrographic studies. So here we just only
But we are doing.
here we took the SRS sensitive strain, then we treated with membranolytic agent, and you can see that they are not damaged. But here these are these are resistant and these are sensitive. The sensitive strains are damaged so much, and this is because of a difference in the lipid composition. So that composition determines their mobility and the rigidity of the membrane. So these are, in summary, the liposomes can be used as a model system to study the membrane-mediated antibiotic resistance in bacteria. Thank you, Bill.
All the best. Thank you very much. Any questions to clarify? Only one? Oh, yes, sir.
Nice talk.
You are just a speaker, is it?
Yeah. Speak, William.
Yeah. Yeah. This was. So what exactly? It's there. This one? Yeah. Yeah. So here we are using NBD labeled lipid. So NBD is there. So we fluorescent here labeled lipid. We can name. No, not the name. Yes. So they are fluorescent lipids. Yeah. So actually here, so here we use two fluorescent lipids. So one is the NBD labeled phosphatidylrhodamine, and another one is rhodamine labeled phosphatidylrhodamine. The rhodamine and they are the good friend. Yeah.
Okay, thank you very much.
Thank you.
So you leave it, or you can get your certificate.
Yeah, please start. Now I would like to request Alok Kumar Pandasar for his oral presentation.
begin. Okay, so good morning to all of you. So I'll be speaking something about the ethnobotanical study of the medicinal plant, which was carried out in Guled Botanical Garden in Ethiopia. So this study started majorly due to the COVID times, as my PhD student went back to his country, and we were able to do some good work. My student actually. He has taken. Okay, so this is the basic of the ethnomedicine to the clinical trials. So what we do is ethnobotany and ethnomedicine knowledge gathering, then the qualitative, quantitative analysis of the data, and then the identification of the traditional medicinal plants used by your healers, and then the phytochemical screening, and then the lead identification, lead optimization, lead development, drug candidates, and then to the clinical. So this is the place. This is a huge place of Guled Botanical Garden, 705 hectares over. So what we started to do is many of the traditional healers from the environments of the GBG as well as from the sub-cities. Now I'd like to say that Addis Ababa is the capital city of Ethiopia. So a lot of people depends on the traditional and the conventional way it is. So the current work is a comprehensive documentation of the ethnobotanical and the ethnomedicinal information that we got from them. It provides an insight into the rich ethnomedicinal knowledge that we have in Ethiopia. I would also like to stress over here is that around 80% of the Ethiopian population do not prefer to go to your modern healthcare. They prefer to go to the traditional healers whenever they have some health issues. So this is the place that we have. As you can see, is around 90% of it, or above than 80% of it, is the protected forest, and then socio-economic and all. So this was the socio-demographical information that we have of the healers. As you can see from here is most of the healers of the age are in between. They are the younger generation. They are not too old. So, and we also see that they already have the modern education with them. That is around 58% of them. Apart from that, also they practice the traditional medicine. So, majorly we found out that most of the plants used belong to the Astraceae family, followed by the Rosaceae, and then the life forms that they used, that was majorly for the herb or the shrubs. So apart from it, we also saw that the plant parts that they highly use is the leaves and the roots. Now I'd like to bring into one thing over here. The more you use the roots, the more the plant becomes vulnerable or endangered, because you have to pick out the whole plant. So leaves is mostly preferred way, but always it doesn't happen. Now apart from this, they use the methods of preparation as crushing and pounding the most, and the topical and the oral administration was majorly the preferred route. Now the ailments, when it comes for the treatment, we found out that the general disease and the skin disease were the majorly treated ailments by the traditional healers. So we also tried to do a similarity index of our data with some of the near about sample areas, and we found out that two places, the Debre Libanos district and the Ankobar, have the highest similarity. Now the first one may be due to the geographical location between the things, and the second one may be due to the ethnic group. We found out the ethnic group in Ankobar and in the place were almost the same. So the summary of what we have till now is, we have 81 medicinal plants belonging to 47 families. Astraceae we have the most, and then we have the plant forms that are herbs, used plant seeds and leaves, and they are mostly done by crossing and the tropical application. Now the skin and the general disease are mostly the treated one. Now one thing I would like to bring over here is, most of the plants that we have over there is endemic. It's only found in Ethiopia only. So 63 of them were there out of 9 and were endangered, and 7 are already vulnerable because of the excessive use. Now, among those, we found out that Echinops cavericomycin has been cited very high, 60 times. And it is not only used for traditional medicine, but they use it for various other things, like getting away the mosquitoes through the smoke, inhaling, and all those things. So the takeaway from this is the E. cavericomycin is an endemic plant and it is already endangered due to its excessive use, and the root of the plant is being used, as I've already told you. So the possibility that this may go extinct is very near because it is only endemic to Ethiopia only. So that's why what we started was we wanted to do some work in order to prove that whatever is there in the roots, does the bioactivity is same in the leaves also. We got this work published here in Frontiers in Pharmacology. So next, our hypothesis was, since E. cavericomycin is used throughout Ethiopia and it is being used in a very high amount, there were various bioactivity studies which has already been done, but mainly confined to the roots. No one carried out with the leaves. So what our literature showed was there were no bioactivities done with the leaves except some discrete study. That also is not very fruitful kind of studies. So we went on to compare the roots and the leaves. So from the roots and the leaves, we saw that the acetone and the methanol extract has very familiar kind of phytochemical profile. And we also did a quantitative analysis of that, and we also found the similar kind of notation what we did in the qualitative aspect. So we went on to do the FRAP assay, and we saw that the antioxidant potential of the roots and the leaves are almost similar. But as you can see, toward the lower concentration in the FRAP assay, in the methanol extract, there is some difference. So we went on to do again the DPPH assay. The DPPH assay, we found that the roots and the leaves have a similar antioxidant profile. We again went on to do the ABTS assay to prove it again, whether they are having it or not. So roots and leaves, again, they showed a similar kind of antioxidant potential. So what we are trying to say is, this study clearly shows that the methanolic and the acetone extract of both leaves and the roots were similar, or at least the antioxidant activities are similar. So what we propose is that, or hypothesize, is that the use of the leaves in the preparation of traditional medicine can be told to the healers to find out the efficacy, which my student is defending tomorrow, his PhD. So after he is going to go to Ethiopia, he is going to try and approach and do that. And let's see what comes out of it. So the forces, so he is the main force behind the work, the major force. These are the supporting forces we have. They are my colleagues and the thinking forces, additional. With this, I end the talk. Thank you.
Any question? The student is Ethiopian or Indian? Ethiopian. It looks like an Indian. Any question? One question, please. Okay, since there is no question, we are passing to the next presentation. Ma'am, you can announce. Now you can announce. So, don't touch. Announce.
The next oral presentation will be a joint oral presentation. So I would like to request Dr. Priya Kumari ma'am and Dr. Vashanti Lakshmi ma'am for their presentation.
So this is the first time. This is the first time in the world 2% are percent. Okay, joint presentation. We hear about that joint project. This is a joint presentation. Okay.
Thank you, sir. Thank you, the organizers, for giving me this opportunity. And dignitaries on the dais and of the dais, dear students, we are going to present today not a research work, but our topic is under social transformation. So we are exactly working, as some people are working very actively in the lab, we are working in the community. To be precise, we both are activists doing a lot of work in the community. So I'm here to present the talk on ecological restoration through massive tree plantation and unique approach by Council for Green Revolution, which is an NGO, and we both are volunteers. We are just volunteers doing work under this NGO who has been established. This was established on the Earth Day on April 22nd, 2010. Since then, the vision of the NGO was to develop greenery in the semi-arid regions of the Telangana state and also Andhra Pradesh. And our vision and mission was to develop this greenery in the area. And our focus was on regreening and improving the degraded land spaces of Telangana, Andhra Pradesh and nearby states. And our initiatives are GRACE, which is called as Greens Alliance for Conservation of Eastern Ghats, one of the initiatives. And second initiative is CELS, Council for Earth Leadership and Sustainability. We'll talk more as the presentation goes on. Our main areas of interest are to organize conferences, seminars, workshops, attend meetings, conduct meetings, webinars on various topics like nature conservation, restoration, climate change, and sustainable development. Our main theme of NGO is several programs which we call as milestones of our program. One is Bangaru Telangana to Balala Harita Haram. It is a Telugu word where Bangaru means it's a golden Harita Haram initiated by the students. That's why we call it as Bangaru Telangana to Balala Harita Haram, where we have chosen as our main pillars of this program are the school children, because we experimented with many people in the society, like we tried to motivate the elders of the society, the youth of the society. But we found that after our research, we found that children were very effective group of individuals where we can choose them and sow the seed of this environmental consciousness in their mind, so that they can take it forward throughout their life. Once sensitized, they are sensitized ever for their lifetime. And that is our investment, where these children can because they are the future builders of the nation, they can take it forward. And wherever they are thinking about environment and our planet Earth. So we started this program, and for this, which we received in 15 and 16, we did with the government program, because as it is the government of Telangana, after the formation of new state in 2014, it emerged as a new state. As the interest state, the government of Telangana wanted to focus on the greening, regreening of the arid, because it's a very dry area. It comes under Deccan Plateau, and it's a very dry region. And most of the places, only few places in Telangana are under agriculture, and because it's desert-like environment, because it receives less rainfall, and most of the places are very dry and hot. So our methodology, because we founded, we were working already in the community. A government observed our work and has invited, and as it is, invite our Harita Haram program into the government program, and it is called as Balala Harita Haram. And next we started another initiative which is called as Kalam Smruti Vanam, in memory of our APJ Abdul Kalam ji. In his memory, we have planted several saplings in so many schools, and Balika Vanams, which are done in the Kasturba Gandhi residential schools, where poor girl children are, or orphan girls
provided with the samplings which are having high nutritional value, and also they were met with the ecological needs of the students. And another program is Smruti Vanam. Smruti is memory. In the memory of the lost, departed people, maybe grandfather, father, brother, sister, whoever it is, in their name, we give a sapling to the family. So they plant it in their premises or wherever they want to, and they name that plant with their beloved person, which is called as Smruti Vanam. Another one is Prakriti Samskriti. Now, as the name says, Prakriti Samskriti. Samskriti is something which is related to our tradition, and because of this Prakriti Samskriti program, we have identified, like we have Bilva Patra. I think if you are Shiva Bhakt here, you will know what is the importance of Bilva Patra. We give most important patra that we give to Shivaji, and during Dasara celebrations, we have this Jammi Vriksh, where they give the leaf of Jammi, which is supposed to be the gold. Yes. Thank you, sir, for correction. And then we also have, in Telangana, there is a festival called Batukama festival celebrated during Navratri days, where they make Batukama with flower arrangements. And these flowers are they are chosen in such a way, very scientific method that these Batkamas, once celebrated, they are put into the waters. So they clean the waters. So that's the concept behind that. Neem tree, banyan tree, which are sacred saplings, they are also distributed in the community. So our approach is, so these are our milestone programs. So our main aim was to do with the children. So how we have done this program, it is by emphasis on the awareness, essay writing, painting competitions, rallies. So this flow chart shows how we are doing. So we also teach the students, we try to tell them the importance of the planet Earth, greening effects, carbon sequestration, etc., all the importance. And also we try, sapling, just giving the saplings and going away is not our work. So we try to teach them how to dig a pit, how much depth and width it should be, and give the sapling so that it is scientifically, the sapling is planted, and survival rate, our survival rate is 90%. And so that we could achieve because of this transition. So one side we have students, and other side we have volunteers who are volunteers like we both who are standing here come under the second group, where we go to the schools, make them aware, try to teach them what it is, and then we give them 5 saplings to each student. 3 are flowering plants and 2 are shade plants. These flowering plants ensure that there is the attraction, as our butterfly man is sitting on the dais. Yesterday we have heard from him what are the importance of the butterflies. So they attract butterflies, pollination, etc., etc. There are many advantages of the flowering plants and shade plants. And this, we don't restrict the students that you have to plant here only. So he wishes to take that plants and he can plant in his house. If he don't have place, he can place it then in his friend's house or any other place in his village, wherever. But his duty is to protect. So likewise, we have given no restriction, but they are enrolled in the school program. We have a manual where we collect all the data, distribute them, and we have the record of all the children who have participated, how many saplings they have given, how many of them are surviving, and where they are surviving. All are geotagged. So this is the picture to share is that Vana Chaitanya movement, where students, after we do our job of encouraging them and providing awareness, they take out a rally, they sing songs, they do whatever, and the small kids, they go around the village. The third one picture which shows they are doing a Gram Panchayat meeting in the village. So when children go and talk about environment, elders will feel ashamed. We are not doing anything. These children are motivated. So let's do. So parents, village sarpanch members, everyone come along and help these little children to whom we have given the saplings and ensure that they are protected and they are surviving. My time is up, but I request the organizers to give me the little more chance, little more time, because this is something which we have done with the community. It takes some time. Yes. One minute. So, these are some saplings in different places. This is Kasturi Balika Kendra. And we have geotagged every tree, and youth, we have taken the help of the youth there. What is the importance of the sapling, what tree, everything we have done here. So you can see different villages, different organizations, and you can see how arid zones, how they are protected and taken. So some NGOs or some organizations also they come. So this is one classic picture where in summer when schools are over and children are at home, in cities they send for some summer camps, some music, drawings, etc. But what we do is we engage these children and we collect the seeds, native seeds, and make seed balls with manure and the mud. And why do we do this seed balls? Because the places where we can't do a sapling plantation, we just go and spread the seed balls, where by a natural mechanism they germinate, and the seed balls are, we found that this was also very successful model, where we have seen the germination of this seed balls. For seed,
Okay. Oh, right.
For seed balls, generally we'll choose the wild varieties of seeds where we can't go and plant, like hilly areas and all those things. We simply take these seed balls and we'll throw. Those are going to be made up of with the along with the manure and all those things. So when the time comes, rainy season, they are going to sprout. So this is the very concept to giving and to attach the students. So madam said that 90% survival rate. How we can broadly say that this is how where we are making bond with the trees. Like Raksha Bandhan, we'll do the cultural part that we are going to do it for the tree. So student Raksha Bandhan. So students are going to be attached to the tree. This they are going to do every year. So this and birthday celebrations, once they whatever saplings they planted, they have to do celebrations for that. So because of that reason, they are attached every year. So then we come to know the survival rate. It's a scientific methodology, attaching sensors and all those things is a very costly process. So we adopted this to know the survival rate. So this is where imparting the plantation in the cultural events as a part of spirituality. Spruti Vanam. So this is the beloved ones. This is how we are going to approach to the students, the light corner, the child, and semi-eritreation. So there also we are improving the green cover. So every school green teacher is going to be there. One of the teachers already they are working. We are going to choose whoever is interested in plantation. We are going to take them as a teacher and they are going to monitor the work. So this is actually Eastern Guard Green Alliance and one of the branch of Council for Green Revolution. So a lot of the flora and fauna is represented. So this book is released by these personalities, great personalities. So where the program is happened, the 2016.
So I'll take another one moment where on the same diet we have conducted a national seminar on the same diet, Utkal University, where Madam and Sir had helped us. A huge round of applause for all the students then and now. Then and now. The same kind of encouragement and enthusiasm I see in the department. Hats off to the zoology department and the students of the zoology department. I bow to all of them personally.
This is the one more organization. Now we are into the leadership programs. So This is the program where we are going to develop curriculum
Ma'am,
for
how
the
much time you can take?
only 1 week.
1 minute. I will count.
Oh, thank you, sir. It's one of the center, generally we call it as art center. Now we are developing sustainable development goals, there's 17 goals. So we are developing curriculum for that. Already we are having MOUs with national institutes, NIT Warangal and other institutions. The sustainable development goals itself one of the goals. Circular economy and environment is the one of the goals. Climate change and environment. So like that we are offering the courses, and even the CELS is providing internship for the students who are interested. Why? Because the CELS is located in the Eastern Ghats. It's one of the part of Eastern Ghats. You can do the research and even you are having accommodation. So why? Because a lot of flora and fauna is available. Why? Because the place is nearby the forest, directly center at center. This is the center. Even it is made of build eco-friendly building, green building, sustainable building. So we can have the comfortably we can have the accommodation for 100 members. This is where actually programs are going to be happen for leadership, for all the stakeholders, from the school children to farmers to grade and it means hierarchy. These are the awards received by the Council for Green Revolution from the state government. This is the ladies who wore pink sarees. So only one single knot. I'm the mother, so I'm taking so much from my mother. I need to protect her. The only single knot made all these things. This is what the work is in the textbook for school children, 5th class. Now it was there, still continuing for these are the publications. Why? Because most of the literature in English not available in local languages. People transform that language. This is how it is relevant to. So generally we, while seeing that it is of NGO work we are presenting, but it is relevant. Why? Because we are the part of biodiversity conservation and as well as economic sustainability. We are restricting environmental alterations and climate change. This is how a number of schools participated, number of schools involved and number of students were participated. With the little student group also, how much plantation is happened. So where the inspiration works literally lot. So this is those only one neem tree is taken. Why? Because we can't scientifically get the data. So only neem tree is taken for the calculation of carbon sequestration. Thank you.
Okay. So that congratulations for
Thank you, organizer, for
one of
speaking.
you. And then we are within time for posting. We are congratulating. We are giving. Okay. Yes, ma'am. Come.
Now I would like to request Asmita Samal for her oral presentation.
Second, we'll break here. We'll take a break here. Yes, Smita will have it after the break. Okay. So we come to the end of the first session of the last day of this 3 days international conference. So let me extend my heartfelt thanks to Professor Murugan for his cooperation and organizing and handling the first session. So in the first session, we had different types of presentations, and the last one, which is what I believe is very important, how to protect our environment, how to protect our plants. So, and two persons coming all the way from Hyderabad. And in fact, in 2016, we came to know about their organization, and they came for the first time to organize any seminars here in Utkal University. So that was the beginning, and I'm happy that we still continue the relation, and hopefully in future this relation will continue, and this plantation program will come up in a better way. Thank you very much. Thank you. So now we'll have a break for tea.